In this video, which is part 2 of the cancer series of 2 videos, I'll be talking about the molecular basis of cancer, talking about some of the mutations, specific mutations, and other things that occur during the initiation and progression of cancer. Many different molecular events occur during the initiation and progression of cancer, and these have been shown in several different, many different studies actually. And that includes point mutations, recombination events, including chromosomal translocations, gene amplification, and the effects of mobile genetic elements such as viruses, transposons, plasmids, and introns. BRCA1 and BRCA2, as indicated in the first video, are involved in the recognition and control of DNA damage. So there are certain sensors that sense DNA mutations and other defects in the DNA, and those signal BRCA1 and BRCA2 to start either going through repair mechanisms, start the repair mechanisms up to repair the DNA, or to signal P53 to send the cell through apoptosis or cell death. And BRCA2 here is located on chromosome 13 up near the centromere, and BRCA1 is over here, located also close to the centromere on chromosome 17. So in a couple of studies, the tumors of the breast cancer tumors were studied as far as their mutations, their point mutations, thinking that, well, if there were some very characteristic mutations in the BRCA1 and BRCA2 genes, that maybe targeted therapies could be developed. So this shows the frequency of mutations in the BRCA1 gene up here and the frequency of mutations in different exons down here in BRCA2. And you can see that there was no clear mutation there that was causing all of the problems. And some of these indicate, so the ones in red, those lines in red, indicate specific mutations in the coding sequences that were more frequent in breast cancers, and the ones in blue, those had to do with intron-exon borders. And in all cases, it seemed like the point mutations were pretty much spread throughout both of those genes, and so there was no commonality with those breast cancers other than they debilitated the BRCA1 and BRCA2 proteins. Recombination is a frequent cause of cancers, and so this is just a diagram showing some of the chromosomal translocations, that is, crossovers between non-homologous chromosomes that are known to cause cancer and sometimes other diseases. So here's a crossover between chromosome 14 and 8, which causes Burkitt's lymphoma, which I've mentioned in a previous video, and then acute myelogenous leukemia between chromosome, a translocation between chromosome 1 and 12, and follicular lymphoma between chromosome 14 and chromosome 18 over here, Ewing's sarcoma, another type of cancer here, between chromosome 11 and 22. There are also some other diseases caused by chromosomal translocations, including schizophrenia here, a translocation between chromosome 1 and 11, and Down syndrome, which I mentioned last time, a chromosomal translocation between chromosome 14 and chromosome 21. This is just another way to look at that same thing. So these are the chromosomes 1 through 23 arranged in a circle, and then the chromosomal translocations are indicated by the lines. So you have a crossover between chromosome 22 and 11, and that causes one Ewing's sarcoma, and then all of these others indicate other types of cancers also. And these are just the cancers are listed here. Crossover crossovers or translocations affecting chromosomes 3, 12, 14, and 22 are the most frequent that are seen in different types of cancers. And the ones in dark gray, that is chromosomes 8, 9, 11, 15, 17, and 18, are also frequent, but a little less frequent than those others. And then going back to these ones in white, those are very infrequently associated with translocations that cause cancers. These translocations often lead to fusion genes, which then produce fusion proteins. So in this case, you have two different genes. You get a crossover within the intron, and that's the most common case, getting crossovers within introns. And then you produce two new genes that produce aberrant proteins that may or may not function normally. Usually they don't function normally. And then you have a second case here where you have a fusion protein which exchanges promoters. So in one case you might have promoter B, which is a very active promoter for this gene here, this upper gene B, and then a promoter C, which is less active. And then you switch those out. So in one case, the protein gets the very active promoter, and in the other case, the other gene gets the rather inactive promoter, and that changes expression of those two genes and their proteins. And here are a few specific examples of those. Here's a crossover here that causes follicular lymphoma. So now you've put a very active promoter next to this sequence over here, and you get aberrant expression of that gene. Another one here, diffuse large B-cell lymphoma, is another one where a very active promoter is put next to a gene that usually is not turned on to the same extent. Here's the Burkitt's lymphoma, which I've mentioned before, which is put next to the MYC gene, which is a transcription factor. So now you're activating that transcription factor more than it's usually expressed. Then here's a prostate cancer carcinoma, where a hormone gene is put next to a very active promoter. So here are some of those too in a little more detail. So here is follicular lymphoma, which is put next to a very active enhancer, and so it also turns on transcription of this BCL gene, which encodes for a protein that is an apoptosis inhibitor. So essentially this makes those cells immortal. Burkitt's lymphoma down here, you put a very active promoter next to the MYC gene, and the MYC is a transcription factor, which will turn on the expression of many genes, so that also increases expression. The diffuse large B-cell lymphoma, also you put a very active promoter next to another BCL gene, which is an apoptosis inhibitor. Again, that makes the cells immortal. Here's a prostate cancer rearrangement, translocation. An active promoter is put next to the estrogen receptor gene, so that increases transcription also. The estrogen receptor is a transcription factor also. So whereas this would be a gene that's normally turned on higher in females, it's still a gene that you find in males, and in this case, you're actually activating it in the prostate. Then there's chronic myelogenous leukemia here. So you, again, have a very active promoter put next to the Abelson gene. And the Abelson protein is one of the proteins active in signal transduction that interacts also with the RAS pathway, which turns on the MAP kinase pathway, which turns on transcription factors. So this is a diagram showing where some of the genes and proteins that have been found in different tumors. These are the ones, the labels in red. So there's RAS, which we talked about before. There's the Abelson that I just talked about. Here's the BCL gene here and here, and those proteins are responsible for delay of apoptosis or an inhibition of apoptosis or cell death. P53 is also one that's involved here in stimulating apoptosis. Here's MYC, which is a transcription factor that increases gene expression. So those are some of the ones affected by these chromosomal translocations that I just talked about. So here's one of those. Specifically, this is a chromosomal translocation between chromosome 8 and 14, forming these two novel chromosomes. And that causes Burkitt's lymphoma, which are tumor cells of B lymphocytes. And just like with point mutations, there are many possible changes that form Burkitt's lymphoma, but all of them involve the MYC gene having a crossover with a different gene that has a very active promoter, an enhancer, and a switch that's on constantly. So here are some of the points. Two are in introns and one's in the exon of this MYC gene, and then this is in the immunoglobulin region. Over here you get a crossover. Also, that one is in an intron. These two are in the switch or the transcription promoter region. And here are some of the mutations that are chromosomal translocations involved in chronic myelogenous leukemia, where you're joining together this Abelson gene with another one, a BCR gene. And the Abelson gene is responsible for part of signal transduction that's close to the RAS gene. And all of these crossover points that have been found are within the intron regions of both genes, and this then creates an aberrant ABL gene and ABL protein, so that affects signal transduction in this case. The chromosomal translocations also affect other types of genes, and in this case, it's affecting a tyrosine kinase domain on one protein, the ALK gene. And so what's happening is normally this protein ALK phosphorylates different proteins, activating them. But when you change the gene, especially the promoter region, then it's turned on to a greater extent. So then you have a lot more tyrosine kinase in the cell, and that will phosphorylate more and more of those proteins, and thus activating them. And that's known to cause small cell lung cancers. Another factor in oncogenesis or cancer formation exists with these genes that have a bromodomain, and the bromodomain of these proteins are things that can interact with histones on the chromosome, and so they can remodel, that is, change the conformation of the DNA so that it often opens up regions for transcription or closes down transcription. So it depends on where they're interacting. Usually they are increasing the transcription of certain genes, thus causing a cancer. So here are the wild type domains in these various proteins, and you can see the bromodomains in some of them. But you can get crossovers between these and other genes and cause aberrant genes and proteins to be formed. This one then has a bromodomain that normally doesn't. Here you're getting a crossover of a bromodomain gene, but then changing the level of expression of that particular bromodomain protein. And there are other chromosomal translocations down here where you're getting a change in the bromodomain genes and then the bromodomain proteins so that expression is changed both in those bromodomain proteins, and when expression is changed there, then that affects chromosomal remodeling and transcription of other genes. And so these are also known to cause certain types of cancers. Crossovers also cause changes around certain genes. So here's the KRAS gene here, and it's small because we're looking at a very large area of the chromosome. So you can get rearrangements along the chromosome, which creates usually higher copy number of regions around or within that KRAS gene. And that includes these head-to-head dimers, so you're getting new sequences added in a head-to-head manner, that is, they face one another, or in a tail-to-tail manner, which is they're reversed on the chromosome from one another. Also, tandem duplications indicated here in green, so you're getting increased numbers of certain sequences around that gene. You can have deletions, which are common in recombination events also. And then interchromosomal exchanges, that is, chromosomal translocations, or just movements of one part of the sequence onto another chromosome, or deletion from that chromosome. So here's another view of how those can happen interchromosomally. You can get a deletion of a region or a duplication of a region. Often these two are related, so you get a deletion from one chromosome and an increase in the other chromosome, and it can be homologous chromosomes or non-homologous chromosomes. You can also get inversion by usually looping around or pairing with the other chromosome in the opposite direction. You can also have chromosomal translocations. Here's an insertion of one part of one chromosome into another. And here's just a simpler translocation here of one end of one chromosome being added to the end of the other chromosome, and vice versa for the opposite chromosome. In this case, it's chromosome 4 and chromosome 20. So crossovers can create quite a few differences among the chromosomes, and many of them can cause cancers and other diseases as well. So you can get crossovers between homologs, either during mitosis or meiosis. For cancer, of course, it would be during mitosis. And although it seems like that wouldn't make a big change, it can sometimes cause a change right at the position of the crossover, so you can get misalignment there. You can also have regions inverted along the chromosomes, and in this case, these inversions may affect regions right where the inversion takes place, those borders. And then you can have misalignment during recombination, adding to one chromosome and deleting from another one, and that can cause actually large changes. And finally, the more obvious ones that you can see cytologically and then see in tumors are chromosomal translocation between non-homologous chromosomes. So these are very common in cancers. It can also lead just to death of the cell, and then you normally wouldn't even detect that. Other changes that are characteristic in many types of tumors are gene amplification. So you get increased numbers of copies of certain genes. So in lung tumors, the somatostatin angiotensin-like peptide receptor. In lung tumors, you get between a 3-fold and 5-fold increase in the number of genes, and that increases the amount of protein overexpression, in this case by 247 times. Here's one in colon cancer, where you have a high number of genes, copies, so the highest fold is 7.6-fold, but that increases the amount of protein, aberrant protein, by 1,000-fold. And then in ovarian cancers, up to a 7.5-fold increase in the number of gene copy numbers and about the same amount in overexpression of the protein. And then in pancreatic tumors, about a 2.5-fold increase in the number of genes. But that leads to about 137-fold increase in the amount of overexpression of that protein. And then down here, here's amplification of the CTSZ gene, which produces cathepsins, which are proteases. That is, they degrade other proteins. So in colon cancer, there can be up to a 12-fold increase in the copy number of those genes, and up to a 52-fold increase in the amount of that protein, cathepsin. In breast cancers, a 5-fold increase in amplification of the genes, up to a 10-fold degree of overexpression of the protein, and in ovarian cancer, up to a 3-fold amplification of that gene, and up to a 10-fold increase in the amount of those proteases. So here are those in graphical terms, just to put it on a different scale, I guess, or a different way of looking at it. So here are the somatostatin and angiotensin-like peptide genes here increased in lung, colon, ovarian, and pancreatic cancer. And then the degree of the overexpression of the protein, that is, the amount of protein. And you can see in colon cancer, it's really expressed to a much higher degree. And then for cathepsin, the amount of gene amplification copy number up to a 12-fold increase in colon cancer. And the amount of protein, and these are proteases again, breaking down other proteins, is really much higher in breast cancer, but it also is high in colon and ovarian cancer, above normal anyway. The amount of amplification of those genes can actually affect survival. And so here, just looking at the HER gene that is involved in some types of breast cancers, if the number of copies of the gene is amplified, usually 2 to 5 times, then the survival rate goes down because the breast cancer is more aggressive. And by the time you get to 10 copies, the survival rate can actually be lower than this. But then with a single HER2 gene, survival of the patients is extended by sometimes quite a lot. So I hope this is leading you to the conclusion that a lot of cancers, or all cancers really, are just a dysregulation in the expression of specific genes, including those that encode proteins for receptors, those that are involved in signal transduction, those that are responsible for control of the cell cycle, control of transcription, control of repair of the DNA, and those that are responsible for either stimulating apoptosis or cell death, or those that are responsible for inhibiting apoptosis. As I mentioned when I was talking about what types of things can cause mutation, I talked about briefly how microorganisms, including viruses and bacteria and fungi, can cause cancers or mutations at least. And so on this top one, you have a virus infecting a cell. It then causes a change in the nucleus, sometimes inserting into the nucleus, and then causing a transformation of those cells into cancer cells. Of course, you can also have viruses that are just lytic, some that cause a permanent infection, and then some like chickenpox, like herpes zoster, that can cause latent infections. That is, they can stay in the cell for some time and cause continuous infections at a later time. But many of the viruses that can cause cancer do it in a way that's sort of general to many types of these cancer-causing viruses, including Bruce sarcoma virus, which is an RNA virus. It carries along with it a reverse transcriptase, then it makes a DNA copy of itself and can copy itself into the genome. When it is transcribed by the host and then makes additional infectious particles, often it will carry along with it an altered host protein or host gene that makes an altered host protein. And those can often cause tumorigenesis and cancer. So in this example, the RNA virus is carrying along a Src gene, which is involved in signal transduction. But it's not the normal Src gene. It's carrying a mutant Src gene with it, which then can interfere with signal transduction in the next cell that it infects. So here is how that occurs with an oncogene and a proto-oncogene. So the host gene is one form, and the viral oncogene is another form. So on top here, the ALV and MULV, these are normal viruses with the GAG, which includes the capsid of the capsid genes and nucleocapsid genes of the virus. POL, which includes the reverse transcriptase, etc., and envelope also, which includes some of the outer proteins of the virus particle. And we've talked about this earlier when we talked about HIV. But then in these viruses that carry oncogenes, they carry along with them the normal gene that was in the host, but it has all of the introns removed. So it's an intronless version of the host genes and eventually the host proteins that they form. So here's the one in the Rous sarcoma virus, RSV, which carries along with it a viral Src gene, which differs from the Src gene in the host, which has 12 exons and 11 introns. In the viral version, it has the 12 exons but no introns. And then there are other examples. Here's a BALB virus, which carries an aberrant version of the RAS gene, and then the RAS protein is aberrant in those infected cells. Here's virus MC29 that's carrying along a viral MYC gene, a transcription factor, which differs from the original host type, etc., other types. Here, where they have the viral oncogene and then the cellular proto-oncogene. That is, it's a normal gene that can be altered to become an oncogene. Here are a few specific examples of viral oncogene for June and the cellular proto-oncogene for June, and then this is the viral oncogene for FOS, which is also a transcription factor, and the cellular version of the proto-oncogene. So these black bars on the bottom show where there are major differences in the protein. So here in June, that intron, which is normally there, is missing. And then over here, there are several sections also missing from the viral version, which makes that into an aberrant transcription factor. And here's the viral FOS gene, which is missing this entire 3' section, which would be then the carboxyl end of the protein is missing, so that it doesn't function as a normal FOS protein in the infected organisms. And again, that those are both indicated in oncogenesis or carcinogenesis. Some cancers also can cause a change in an RNA, a microRNA, that can then inhibit the expression of some of the cellular RNA. So in this case, there's a DNA virus. It forms in this case, a PRI microRNA. Then you have that being processed, becoming duplexed with the risk elements, and then actually causing loss of expression of certain mRNAs that are already out in the cytosol. So again, this can also create a situation where a tumor is formed and a cancer is formed, and causes actually sometimes the cell to experience other mutations also because of that. So for these viruses, there are actually two different mechanisms for affecting transcription of a proto-oncogene or an oncogene, and one is on the top here. So the virus infects the cell, picks up a proto-oncogene, which it actually makes into the oncogene by mainly deleting the introns, or taking an RNA version of the oncogene and reverse transcribing that so it contains no introns. And then infecting other cells where it carries that oncogene with it, converting those cells or transforming those into cancer cells. Another mechanism is that they often have very active promoters. So they will integrate next to a proto-oncogene and turn it on to much higher levels, such as, turning on MYC or FOS or JUN or something like that, and affecting increases in transcription that way, just by inserting a promoter sequence next to a proto-oncogene, turning it into essentially an oncogene situation. In the case of human papillomavirus, shown here, it's a double-stranded DNA virus rather than an RNA virus, and how it replicates is it moves to the nucleus and makes many copies of itself. Some of the copies actually get integrated into the chromosome, and so it transforms the cell into a cancer cell. And then those cancer cells divide, carrying the virus along with them, and ultimately then they can metastasize as well. There are vaccines against this virus because it's actually in high prevalence in uterine cancers and other types of cancers. So before people become sexually active, usually they're given a vaccine, and this blocks replication of the virus and infection by the virus, so that you can essentially avoid this type of cancer by becoming vaccinated. Another factor that can influence either initiation or progression of cancer are steroid hormones, including progesterone, estrogens, testosterone, etc. And so here are some of the pathways indicated, all starting from cholesterol. Each one of the numbers in the circles indicate a different enzyme that's involved in those reactions. So over here, you can, from pregnenolone, you can get progesterone, and from beta estradiol down here, you can get estriol. So some of the main ones are here: testosterone, cortisol, and progesterone, as well as estradiol or estrogen. Estrogen is a bit unique among hormones in that it can go through the cell membrane without having a receptor, and the receptors are actually internal within the nucleus, so it moves directly into the nucleus. And so up here you have a ligand-dependent transcription activator. So there's the estrogen receptor. You attach estrogen. It attaches to the promoter region and stimulates transcription of the genes. Also, there can be tethered effects also, so you can have other transcription factors that attach to the estrogen receptors that then stimulate transcription of that gene. There are also non-genomic effects of estrogen. And so those occur out in the cytosol. Those are less well known, but you can have estrogen receptors with or without the estrogen attached, and those can cause physiological effects out in the cytosol. And then there are ligand-independent effects. So you can have growth-stimulating signals affecting estrogen receptors, and estrogen does not even have to be attached, but you can phosphorylate the estrogen receptors, which then become transcription factors and will attach to the promoter region and stimulate expression of that gene. Estrogen can affect cancers in two different ways. So in one way, you can already have some cells that already have mutations in them that will lead to cancer, and then estrogen can actually stimulate those cells to multiply and become larger tumors. It can actually cause changes in other cancers also, not only uterine cancer. So it can stimulate the growth of already pre-existing small cancers and make them much larger to grow much faster. It can also stimulate cell division, which will stimulate also mutation, because you have more mutation every time you replicate a cell. And so that will increase the chance of having a spontaneous mutation, and that can also lead to tumorigenesis and oncogenesis into a cancer. And you might think this affects only women, but it doesn't. So yeah, you can affect breast cancers and uterine cancers because fatty tissue produces more estrogens, so that can stimulate a tumor. The ovaries also produce estrogen, so that can induce a cancer. Growth hormones, such as those coming from the pituitary, can enhance cell division by acting on the estrogen receptors, and then the placenta also forms hormones that can increase cell growth and division. Again, fatty tissue can produce more prolactin, which also is a stimulator of cell division. And some plastics and other compounds, and even beer, can contain some estrogen. And especially now that birth control pills are used so extensively, and much of the estrogen that's in those pills can go down the drain with the urine, and then can be picked up later by a water system. And so people can actually drink small amounts of estrogen in their beverages as well. And then there are things that plants produce that are called phytoestrogens. They're not really estrogens, but they simulate estrogens in the estrogen receptors, so those can also affect this. So some of the effects of prolactin also. So prolactin is produced in the pituitary, in adipose tissue, and by the placenta. So that will stimulate, of course, milk production in the females. But it also has other effects. So the prolactin can affect signal transduction in many ways, and increase motility of the cells, survival of the cells, that is, make them immortal, proliferation and differentiation. So if you already have a cancer cell, or in some cases to initiate a cancer cell, prolactin as well as estrogen can affect those different types of cell characteristics in tumorigenesis and carcinogenesis. So just to sum up here at the end, I talked a bit about point mutations being important in the development of cancer, and pretty much randomly appearing in the genome and in certain genes. And then recombination events, many of those can cause cancers, including chromosomal translocations, which are have been identified as a major cause of some types of cancers. And then gene amplification is important in whether that cancer becomes more dangerous or not. And then mobile genetic elements. I talked mainly about viruses, but also transposons, plasmids, and introns can affect tumorigenesis. And then a little bit I talked, and I haven't got it on this list, but about how hormones, especially estrogen and prolactin and likely some others, lead to, if not the initiation, but the progression of the cancers. So here are some of the changes in cancer rates in the U.S. between 1988 and 2008, and they've changed a bit since then, but not a great deal. So there have been a decrease in certain types of cancer, including stomach, colorectal, ovary, lung, etc. And a lot of these are from better diets in some cases, stopping smoking, and lowering of alcohol consumption, things like that, as well as increased detection of precancers that have helped in surveillance of some of these different types of cancers. And however, there have been increases in esophagus, pancreas, prostate cancer, some lymphomas, melanoma, and thyroid cancers. Thyroid is probably related to radioactivity. Due to radioactive tests and increases in use of radiation for different purposes. Melanoma definitely related to sun exposure, people tanning booths, things like that. Some of these other ones, pancreas and prostate, no one knows exactly why some of those are increasing, but maybe one of these days we'll understand why some of those types of cancers are increasing. One problem with those is the prevalence in some of these, like pancreatic cancers, is low, so it's difficult to look at any particular pattern, even though they are increasing in frequency. So this is the end of part 2 of the lectures on cancer, of a 2-part series again. Okay, so I'll talk to you later in the next video.